Good afternoon. This is Dr. Dan Guerra from VeraMed Studios, recording from the Pacific Northwest. Today I would like to talk to you about a very interesting topic to a lot of people. How does the ketogenic diet work relative to some disease states? Now, today I'm going to take a very small subsection of that and describe how ketogenic diets have been used clinically because of subclinical experiments initially for people that have epilepsy, particularly uncontrolled seizures, and how diet may ameliorate those seizures. So let's just get right into it so that we don't have to waste time with describing this entire background. Okay, just wrong one open. Okay, here we go. Slideshow. From the beginning. Close this. Okay, again, it's the VeraMed lecture, and what we're going to talk about today is, does high lipid diet, particularly a ketogenic diet, correlate with glucose-mediated excitation of hippocampal neurons? Now, why is that significant? It's because the excitation of those hippocampal neurons, as a part of the brain, the hippocampus, particularly the CA1-3 region of the hippocampus, is directly associated with seizure. Okay, as in epilepsy. Okay, so that's me, Dr. Dan Guerra. This is my personal email address. Please contact me for any and all questions about my VeraMed lectures and anything about VeraMed in general or specific. Both necessary and universal questions are, of course, well received. That's my Facebook page. Please take a look at it for all of my VeraMed lectures and a lot of other information about me, about the company, and of course the company itself. There's our website, and there is the VeraMed email address. Contact us in any of these ways, and we'll get back to you, and we'd be glad to have you as a client. All right. So what's this paper about? It's a paper published in Journal of Lipid Research in 2014. What it basically says is that a high-fat, low-carbohydrate diet, which are also known as ketogenic, here called KD, is a current treatment for refractory epilepsy. Now, that was back in 2014. It's still the case in 2018. KD limits blood glucose and produces ketone bodies from beta oxidation of lipids. So that's the key feature there. What ketogenic diet means is that high-fat diet, that is the intake of fatty acids from triacylglycerol, will allow those fatty acids to be preferentially beta oxidized in the hepatocyte in the liver. And then the production of ketone bodies, which is acetoacetate and beta hydroxybutyrate, will allow for the utilization of those ketone bodies in the brain, particularly in the hippocampus, as we're describing here right now, alternatively to glucose. So there's two things going on. One is that ketone bodies are preferentially used in the brain for bioenergy. And secondarily, that glucose is not introduced into the system. So you have to have low blood glucose. Now, that's a bit of a problem if you have gluconeogenesis happening in the liver, which can be coincidental to a ketogenic diet. So there has to be control of blood glucose as well as the production of ketone bodies. Now, that's the key clinical feature that we won't get into today, but I want you to keep in mind as a reference. So this paper goes on to examine the interaction between altered glucose and the neural effects of ketogenic diet in rodents that were fed such a diet. Seizure-prone CA3 region of the accurate hippocampal slices were examined, and those slices from KD-fed animals were sensitive to small physiological changes in glucose. And this experiment is going to be between 3 millimolar and 11 millimolar, which is a good dietary range, actually, in mammals. Anyways, they show a reduced excitability, that's a good thing, and a reduced seizure propensity. So reduced excitability required dietary reduced glucose. Now, I said that. Enhanced glucose sensitivity and reduced excitability were absent in slices obtained from KD-fed mice lacking the adenosine A1 receptor. Now, there's a new feature. We're going to find out that the adenosine receptor plays a key role in this ketogenic diet. Hopefully, I'll be able to explain that to you and provide evidence even from this paper. So the study suggests that the ketogenic diet sensitizes glucose-based regulation of excitability via purogenic mechanisms in the hippocampus. What's the background? Now, I'm going to get very technical right now. There's a protein called pannexin, called PANX1, and that is a hemichannel, and it works in concert with extracellular adenosine triphosphate, that's regular old ATP, and a suite of purinergic receptors. Purinergic receptors specifically, these are going to be receptors that utilize adenosine. So, there are two purines, adenosine and guanine. It's going to be the adenosine purinergic receptor we're going to be describing today, those associated with adenosine, which is an ATP. And those are involved in several physiological and pathological conditions. We're not going to get into all of that, but the pannexin is a player. The pannexin-1 hemichannel, extracellular ATP, and purinergic receptors are involved in cellular activation. They're involved in also apoptosis, stress signaling, and indeed, in some systems, secretion of pro-inflammatory cytokines. So more on pannexins. PANX consists of cytosolic N-terminal domain, 4 transmembrane spanning regions with 2 extracellular loops, and a cytosolic C-terminal domain. Okay, so it's a complex intramembranous protein complex, which functions to correlate intra and extracellular activation potentials. PANXs form large pore channels, in fact, located in that plasma membrane, and they're known to open during membrane depolarization by changes in intracellular calcium signaling. This is very common, actually. Also vasodilation, vasoconstriction, taste sensation, airway defense, learning, memory, cellular differentiation, programmed cell death, as we've mentioned, and during innate and adaptive immune responses. So these PANXs are all over the map in terms of normal physiology and pathophysiology. So they're well studied. So there's a PANX to purinergic receptor signaling. Upon opening of these hemichannels, which is what the PANX are called, I won't go into detail why they're called that, but suffice it to say that hemichannels are a specific subspecies of membrane-associated channels. Anyways, upon opening of these PANX hemichannels, small signaling molecules like ATP are released, and they're released into the extracellular space. Those then can be metabolized, that is, the concentration of ATP can be metabolized, and then signal through a series of surface receptors, many of which are these purinergic receptors. So you release ATP after opening the PANX, ATP leaves the cell, it's metabolized outside the cell, and then those metabolites bind back to that same cell surface, or self-same surface, and then induce via purinergic reception a whole series of cascades within those cells. This is all happening in the CA3 region of the hippocampus, of course. So let's talk about the purinergic receptors. They're divided into 2 groups. The adenosine receptors, or ARs, and they're for adenosine, and then the P2 receptors, which have phosphate, ATP and ADP. P2 receptors are divided into ionotropic and metabotropic. So there's P2Xs and P2Ys. So you see all these subclasses. You don't need to worry about all of them right now. I'm just giving you this detail so you'll know. The P2X receptors are ligand-gated ion channels, and they form trimers. The P2Ys are G protein coupled receptors, which I've talked a great deal about in verified lectures. Just so much, right? Purinergic receptor signaling is fundamental in many cellular processes. So we talked about the PANX, now we're back to purinergic. What are the purinergics involved in? Holy smokes. Platelet aggregation, exocrine, endocrine secretion, those are hormones, endothelial-mediated vasodilation, such as associated with nitric oxide, nociceptive mechanosensory transduction, neuromodulation, neuroprotection, cell proliferation, differentiation, migration. You can think of a lot of diseases, I'm sure, there, you docs. Embryological development, wound healing, inflammation, and cytokine secretion. So just a whole host of, again, normal physiological and pathophysiological states, purinergic receptors involved. So upon release of ATP into the extracellular space, several enzymes degrade ATP, first into ADP, then AMP, and then ultimately to just adenosine. And all those, as I said, signal through these purinergic receptors. There you go. So you've got pannexin opens up, it releases ATP, boom. ATP is hydrolyzed to ADP. We remove the gamma phosphoryl. Now the beta phosphoryl to AMP. Now the alpha phosphoryl to adenosine. All of these have their own receptors. There's the P2X, P2Y, and the AR, right? So all of these are going to bind back to these receptors. ARs are the adenosine is ligand and agonist, okay? So that's the AR receptor. All right, stepwise ATP hydrolysis. Now the mechanisms which limit blood glucose, such as fasting or a ketogenic diet, have been known to reduce epileptic seizures. So this is just something we know. This is, in other words, when you formulate hypotheses, reductio hypotheses, when you're trying to design experiments, which is what we're doing here, you have to deal with some givens, right? You have to deal with the summation of two previous experiments. You have to carry out a dialectic via the scientific literature, right? So what you've got right there is a conclusion, right? And the conclusion is, it seems like when you limit blood glucose, this is the result out there in the literature, when you limit blood glucose by fasting or by a ketogenic diet, it's been shown that you can reduce epileptic seizures. Full stop, that's shown. Now, metabolic changes accompany numerous central nervous system disorders, and the purines, ATP, and adenosine itself are poised to translate cellular energy into altered neuronal activity via bioenergetics. So not only do you have these as signaling molecules, remember this is part and parcel. This is home run. This is where it's at. This is within the wheelhouse of bioenergetics, right? And bioenergetics, again, has to do with ATP. ATP hydrolysis drives all of the energy metabolism in the cell, along with GTP hydrolysis, but mostly ATP. And I just wanted to remind everyone, I know this is in your mind all the time when you're taking long walks, for example, or when you're having a cup of coffee, or when you're watching your favorite documentary on pre-Soviet Russia, right? Those kinds of things, which I know are common to people. The energy charge is how you link how much ATP, ADP, and AMP are in a cell. And the energy charge is just the amount of ATP concentration plus half the ADP divided by all of the ATP, ADP plus AMP concentrations. Okay? So this is just the way that you describe how much energy is in a cell. Now, high energy charge, that means when you have a lot in the numerator, which means you have a lot of ATP and ADP, so you have a lot of energy potential or high energy charge, it shuts down glycolysis. Now the reason that is because you get a lot of ATP, higher in the numerator, and that blocks phosphofructokinase 1. Okay, so that enzyme, when that's blocked, you shut down glycolysis. Now, why is it significant? It's significant because glycolysis is a major anaerobic utilization of glucose. It's how you enter glucose into metabolism. Once you make pyruvate, if you've got plenty of mitochondria and molecular oxygen, you can go all the way through the TCA cycle and make all those 38 ATPs from burning one glucose. But glycolysis is going to give you 2 net ATP, but it's always happening in all cells. But glycolysis itself, front-ended, is inhibited by a high energy charge. In fact, this is also known as the Pasteur effect. Yeah, Louis Pasteur figured this one out. Amazing, I know. Okay, so that's an important issue. So you see how these purines, particularly ATP, AMP, ADP, and adenosine itself, can feedback regulate carbohydrate metabolism. This is significant, okay? All right. Now, here's why. Alterations in metabolism induce a purinergic autoregulation of hippocampal CA3 pyramidals, the type of cells in the hippocampus, neuronal excitability. Yeah. During conditions of sufficient intracellular ATP, reducing extracellular glucose, okay, so what we're talking about here, remember, lowering glucose, induces that pannexin-1 hemichannel mediated ATP release. So you're releasing ATP when you get a reduction in extracellular glucose. That happens with a low carbohydrate diet, for example, or a high ketogenic diet, for example, right? Or fasting. This extracellular ATP, as we've shown now in slide manifestation, is dephosphorylated finally to adenosine that activates neuronal adenosine A1 receptors. We just saw that. And unexpectedly, it hyperpolarizes neuronal membrane potential via an ATP-sensitive potassium channel. Now we're talking about yet another player here. We're talking about an ATP-sensitive potassium channel. And we're getting hyperpolarization neuronal membranes in the CA3 section of the hippocampus. And we know that this is involved in seizures. So that constitutes, as we've seen in the previous slide, an autocrine. That's when you release a substance and you utilize that substance in the same cell via receptor-mediated activation, endocytosis sometimes. It's an autocrine activity. This constitutes an autocrine regulation of neuronal excitability via ATP and all of its family, and as adenosine included, in a seizure-prone subregion of the hippocampus. Boy, that just kind of sets the stage, doesn't it? There is therefore a relationship between decreased glucose and increased seizure threshold. Again, full stop, makes perfect sense, right? Now, by establishing a neuronal ATP release via the pannexin hemichannel and hippocampal adenosine A1 receptors, which have to be in place, coupled to an ATP-sensitive potassium channel, which finally controls that polarization of the neuronal membrane as the final step, right? There is a relationship between metabolism and neuronal activity. Well, wow, who wouldn't have thought that? As a biochemist, it makes perfect sense. I love the linkage, right? Because you're utilizing, remember, these adenosines for something different than energy here once you leave the cell. Right? So the concentrations are totally different than what you see in bioenergetics. Yeah, so here it is again. Again, I got this from a paper published in 2014, Frontiers in Physiology. Okay. Again, you're just looking at exactly the same thing. You're looking at ATP moving through pannexin, the PANX, right? And then stimuli results in the opening of that PANX channel. You release the ATP. You run all these different adenosine metabolites through their own receptors. And then ultimately you have to link this to that potassium ATP channel. Okay. Now, here's data from the paper, okay, the JLR paper we're talking about. That's the Journal of Liver Research, another exquisite journal, by the way, within the family of great journals like the JBC. So when glucose is reduced, so we go from 11 to 3 millimolar. I'm not showing you all the experiments, just only showing you a few of them, data that is from the experiment. I'm actually showing you the experiment. I don't have a movie of that, sorry. When glucose is reduced to 3 millimolar, a reduction in biculene-induced excitability of a keto diet-fed rats was observed. Now, this biculene-induced excitability, that's just a drug that induces excitability of these neurons, okay? So when you have a ketogenic diet, you reduce that excitability. That's a good thing, because reducing the excitability that was induced by biculin means that you're reducing the potentiation of seizure, okay? So at low glucose, this is just the data that, I told you this in narrative form, now I'm showing you the data. Okay? This is over time. This is just an excitability curve. Controlled diet versus ketogenic diet, you get less excitability in that membrane when you have low glucose with a ketogenic diet. And that's just shown here in histogram, right? Less and statistically significant. That's what this is telling you. Okay, now, another really important experiment. I'm only showing you two, the results of. When you antagonize the adenosine receptor, remember that's the last one, the one that picks up adenosine, or you knock it out, as shown here, knocking out, remember, homologous recombination mediated complete ablation of a gene in rodent models. It's a common way to study the activity and the potential utilization of a gene product in a cellular or whole animal system, okay? Using homologous recombination, negative selection, the kind of things I've talked about before and I've been involved with at the research laboratory. Okay, so if you antagonize it, now this is using a drug, this is DPCPX, different drug, right? In the presence of even the ketogenic diet, right? Even in low blood glucose, look, you get no effect on reducing that potential, that hyperpolarization. Now you do, this is the same effect you saw before, but it looks flat because you're not comparing it to a control system. You're comparing it here to inhibiting, right? So if you have no antagonist, you see this reduction, right? And that's a good thing. That means lower potential for seizure. But when you add this drug, which antagonizes the adenosine receptor, wow, ketogenic diet or no ketogenic diet, no good effect even in low blood glucose. Now that is recapitulated, okay, when you look at a knockout. I told you homologous recombination mediated knockout of what? Knockout of the A1 receptor, adenosine 1 receptor. You look at wild-type mice and you look at the knockout mice, and you see that you get a lot of excitability in the knockout mouse, right? And it's the same as if you use the inhibitor when you have the low blood glucose ketogenic diet. So that means that all of the activity of the ketogenic diet is funneled through the movement of that A1 receptor. That's what this is telling you. Using chemical antagonism of that receptor, or you're knocking the receptor out completely. This is its classic molecular genetic type of experiment when you're honing in on one particular gene product. All right, so let's carry out the dialectic very quickly here. So the elevation of adenosine and ketone-induced activation of the adenosine 1 receptor could explain the ketogenic diet anticonvulsant anti-seizure activity. Looks like it, at least. Indeed, agonist-linked A1, that is adenosine 1 receptor activation, is effective in controlling pharmacoresistant seizures. So we have drugs that agonize that site, okay? And when you do, you can control even people who have seizures that are resistant to normal pharmacological therapy. It's very significant. The ketogenic diet is by definition low in glucose, of course. It also has a low glycemic index, of course. So the anticonvulsant mechanism, which involves reducing the excitability of seizure-prone area via pannexin 1 channels, adenosine A1R receptors, and ultimately these potassium ATP channels, could be the mechanism whereby diet controls epileptic seizure. Okay? It's a mouthful, but that is what we've just shown. We're verifying the evidence here, and that's what this paper suggests. Ketone body availability to the hippocampus, therefore, would, okay, here is biochemical understanding. The dialectic is now in full movement mode. Ketone body availability to the hippocampus would, from the diet, would block the effects of glucose by generating its own ATP and the adenosine molecular family. Okay, because ketone body utilization in the brain will go right into the TCA cycle, make NADH and FADH2, and there's where you're going to get your ATP. Not from glucose, not from glycolysis, which is going to be shut down, thank you, because of the high energy charge induced by beta oxidation, okay, and induced by the TCA cycle. Full stop. So blood glucose is correlated with seizure. We already know that, okay? So anticonvulsant effect of the ketogenic diet in vivo reverse quickly upon glucose injection. That's also well known. And it's also well known that when you ingest a high carbohydrate diet, you also lose the anticonvulsant effect. So all of these come together, right? That is a good summation, a good ending of the dialectic, a good relationship correlative study that suggests that ketogenic diet working through the purinergic receptors because of the utilization of ketone bodies in the brain and the suppression of glucose in the serum because of a low glucose diet, and again, this potentiating effect of keeping gluconeogenesis low, because you're going to probably need to do that too. Low circulating blood glucose. This is probably one of the ways the ketogenic diet works to reduce seizure. All right, now let's take a look at this. Now, this paper, published a few years later, more recently in 2016, okay, in Frontiers in Molecular Neuroscience, right? About a year ago, right? Yeah, a year and a few months ago. What are we looking at here? Ketogenic diet. You got these seizing mice, right? And they're fed this ketogenic diet. Funny, I think, the way they draw these things. You make a lot of ketone bodies. You have low glucose. The low glucose allows for this PAN-X to open up because high glucose shuts down the PAN-X, okay? That's one of the molecular ways to control PAN-X. High blood glucose, PAN-X down, ATP is not released from the cell. But glucose is low, PAN-X is open, ATP goes up, adenosine goes up, binds to its receptor. It activates the potassium, outwardly vectorial potassium channel, and causes hyperpolarization, and that gives you the anti-epileptic effect. Okay. So increased ketone body levels and reduced blood glucose levels. Ketone bodies increase intracellular ATP. We already said that. How does it do it? Because when you break down ketone bodies, make NADH and FADH2 in the TCA cycle, and those get reoxidized in the electron transport chain within the mitochondria. And because of oxidative phosphorylation linked in complex 5 of that pathway, you make ATP. Increased ATP, that's all right there in those 2 words, increased ATP, is released to the extracellular space via opening of the PAN-X, right, because of low glucose. Activated adenosine A1 receptors open the ATP-sensitive potassium channel, boom, and that hyperpolarizes those pyramidal neurons, and that's what you want here. Hyperpolarization reduces neuronal hyperexcitability. You got to understand that hyperpolarization can reduce neuronal hyperexcitability. Okay, 2 hyper words, but that's how it functions, okay? And when you reduce neuronal hyperexcitability, which was remember those CA3 slice experiments? That's what we were talking about there, those traces we're looking at, that area under the curve, you reduce seizure. All right, so that's all I want to say today. Again, I'm Dr. Dan Guerra, and we are VerEvMed. There is my Facebook page, which has all of my other previous videos. There's my particular and specific email address, which I give out because I know people contact me through this method. There's the email address for our company, and there's our website. Please contact us. We'll be glad to take you on as a client, where we can answer using verification of evidence in the previously published scientific literature and up-to-date literature of what your particular research interest is. Be you a doc, a pharmacologist, a pharmacist, or a layperson that wants to know something about what is involved in their normal health and also in possible pathogenic systems and diseases. We'd be glad to cooperate with you and your physician. Thanks a lot for your cooperation here in listening to me. So let's go ahead and shut this down. Okay. Again, thanks a lot for your undivided attention. And until next time, bye for now.